Hi, this is Dr. Marcia Anderson with the Center, and I have the privilege of being your moderator for today's webinar. The views expressed by our presenters are for educational purposes only and do not represent the official views or positions of the EPA or the presenters' organizations. This webinar is part of a series providing information on the importance of integrated pest management, or IPM, to community health and the information needed to establish and grow effective IPM programs. Protecting the health of American citizens is important to the Environmental Protection Agency. Children, the elderly, and health-impaired persons face increased risks to their health when they're unnecessarily exposed to pests and pesticides. For this reason, the EPA recommends that all schools, childcare, healthcare, and community centers implement programs that promote the safe use of pesticides and integrated pest management. Effective pesticide safety and integrated pest management programs address not only the safety concerns of using pesticides, but also the causes of why pests, like mosquitoes, may frequent around your facility. As a result, IPM provides all people an effective and sustainable alternative to pesticide-centric methods of pest control. At this time, I'd like to introduce our presenters. Introducing the topic will be Dr. John-Paul Mutabe. He's a research entomologist at the Center for Disease Control and Prevention located in Colorado. Dr. Mutabe has played major roles in responding to vector-borne disease outbreaks, especially West Nile, Eastern equine encephalitis, chikungunya, and the Zika virus. Next will be Chuck Lubileski, Master of Public Health, works for the Maine Medical Center Research Institute in Scarborough, Maine. He conducts research on mosquito host and habitat requirements and the spread of vector-borne diseases. He also is an expert in IPM approaches to reducing vector-borne diseases. Following Mr. Lubileski is Dr. Kathy Murray, who works for the Department of Agriculture, Conservation, and Forestry in Maine. Dr. Murray oversees IPM activities and provides pest management expertise in agriculture and non-agricultural settings, including ornamental horticulture, livestock and poultry, and public and community health. Wrapping up the presentation is Mr. Martin Renya, who's the Technical Operations Manager for the Mosquito Testing Program in Harris County, Texas, or Houston. He's responsible for the in-house virology lab testing program and for establishing the current St. Louis encephalitis, West Nile virus, dengue, chikungunya, and Zika surveillance and testing programs. Our first presenter, Dr. Jean-Paul Mutabe, will discuss the principles of vector-borne disease surveillance and why mosquito monitoring is a key part of mosquito IPM. Dr. Mutabe, take it away.
Good morning, everybody. My name is John Paul, and thank you very much, Marcia. In this short period of time, I'm going to talk a little bit about the principles that govern mosquito surveillance. Now, to begin with, mosquito surveillance does not stand on its own. It is part of organized vector-borne disease surveillance. It is one of the portions, one of the parts, and it is an integral part of it. Now, the vector-borne surveillance as such includes monitoring, organized monitoring of vector populations. That when I talk about vectors here, I'm talking about mosquitoes, ticks, sandflies, and other arthropods. Monitoring level of pathogen activity within the vectors, and infections, monitoring level of infections, vertebrate hosts, including humans, weather conditions, and any other factor. Now, the entire aim here is to predict changes in disease transmission dynamics. In other words, beginning with zero transmission outbreak status. Now, the principles, there are some four principles that tend to govern mosquito-borne disease surveillance. The first one is that there are some of those factors in nature that tend to precede all mosquito-borne outbreaks. This includes like mosquito abundance. That is why we monitor mosquito populations. At times it is weather conditions, and in many cases it is rainfall, hot, dry weather. We know that how dry weather tend to precede West Nile outbreaks. And we are going to look at I'll give some examples about where rainfall can be one of the factors as well. And the second one is that the closer that factor is to the event, the higher the predictive value. In other words, probably the best indicator is having human cases, but that is what we are trying to prevent. And this what actually shows here, what I'm trying to show here is that it is kind of not a really convenient situation because you need some lag time so that you prepare to prevent the outbreak. Now, the surveillance systems must be based on really good knowledge of the transmission and maintenance cycles and the epidemiology of disease of interest. In other words, it has to be based on sound scientific knowledge. Also, the programs depend on sound sampling methodology. The success of any mosquito burst surveillance depends on sound sampling methodology. In that case, we are talking about using the appropriate traps type in the appropriate way. I'm not going to go into details in that. I'm going to leave it to Chuck, who speaks next, to go through some of the examples of how appropriate trap selection is used for the different outbreaks, different disease outbreaks or vector-borne disease outbreaks. And also, program success depends on good identification, solid identification techniques for the mosquitoes and for the pathogens. Now we are still on the subject. There is usually a tendency of using this, of thinking that you can use the same method over and over again. No, there's no such a thing as a universally applicable mosquito surveillance system. The system is always tailored to the disease we are talking about or the disease of interest and to the ecology where you are. So there are variations that take place because there are changes both in the transmission dynamics by vectors caused by different climates. The vectors change according to the disease of interest. Now, there was a question that was posed about what is the role of, say, the state and what is the role of the local mosquito control district or the local health district. Now, when you're designing mosquito surveillance program, it is always tailored to that area where you are. So it is at the local level. It's quite a bit of variation of how states get involved in mosquito surveillance programs throughout the United States. Some of them, they are pretty much left to the local mosquito control districts, and others, the state gets actively involved. But either way, the programs that are being maintained or put together for mosquito surveillance are specific for the specific areas that monitoring is taking place. Now here I've got some examples of early warning signals. For example, there is one where it was noted that by Grady and others in 1978 that whenever there was more than 20 rainfall for 2 consecutive years, that was above the average by more than 20 centimeters. That tended to indicate there was going to be an outbreak of eastern equine encephalitis in Massachusetts. And I'll come back and talk about that later. Also, something else that is usually not monitored is the trajectories of the wind, because they are associated with some of the outbreaks or the positive mosquitoes were detected. That is between 1980 and 1985. Again, I'll come back. Now, this is the first example I was going to give. Now, this shows what happened this was showing what rainfall patterns between 1950 and 1975. What it shows is that there was elevation of rainfall from 1953 to 1955, and it was above 20 centimeters. And beginning in 1955, there was an outbreak of eastern equine encephalitis in Massachusetts. Also, there was another one. There was an elevation from 1973 to 1975. From 1973 to 1974. And beginning in 1974, there was an outbreak of eastern equine encephalitis again in Massachusetts. This kind of model, unfortunately, only works in Massachusetts. It gives you that idea that there was some weather pattern or a weather incident that precedes the outbreak of eastern equine encephalitis in Massachusetts. Another example here, this was showing trajectory of the winds. And there are 2 examples here where you see that from western Kentucky, and this is the study that was it took place between 1980 and 1985 by Sellers and Marouf. And it shows that there was some cold fronts. When cold fronts were coming in, there are some winds that were moving up north, and the assumption here is that it was taking eastern equine infected mosquitoes with it. There are 2 patterns of it. One from western Kentucky ended up with an outbreak in Michigan, and one from North Carolina went northwards and ended up in Maryland, New Jersey, and New York. All of these were not really outbreaks of Eastern equine encephalitis in humans, but positive mosquitoes were detected in those areas, and the only explanation was that northward movement of the winds. In other words, it was the southerly winds that were the only predictive factor that could explain why those positive mosquitoes or positive horses or human outbreaks were taking place. Now, the principal functions of the mosquito-based surveillance program are basically 5: collecting data on adult mosquito population parameters, because whenever you're doing that kind of surveillance, you want to know the species composition, because not all mosquito species are vectors. Some of them are vectors, some of them are nuisance. In other words, they irritate you, but they're not transmitting diseases. The relative abundance, that is always very careful, very important, because usually high abundance tend to precede outbreaks. Now, usually the word density is used, but that is not in the strict sense, because density is usually the number of individuals per unit area. Mosquitoes, mosquitoes like all other arthropods, they don't tend to exist in that kind of parameter. They tend to go up and down. They are explosions and crashes. So that's why we use relative abundance. In population dynamics, that's exactly what I'm talking about. Movement, the explosions and crashes. Now, for some programs, they go much further. They look at age structure and a lot of other parameters, because age tends to be important in the transmission dynamics of diseases. The older the mosquito, the higher the chances they've been infected. Now, the second aspect of this, the second function, is estimating infection rates within the vector itself. Been some traditionally used measures or traditional indices that have been used, including the minimum infection rate or the maximum likelihood estimate. But me, I always recommend using the vector index, because both the MIR and the MLE are a little bit flawed. In other words, you end up estimating the number of infected mosquitoes per thousand. Whereas in many cases, you may not have, in some cases, you may not have 1,000 mosquitoes in an area. So if you have 2 mosquitoes and one is infected and one isn't, then you end up with 500 mosquitoes infected per 1,000, which kind of misrepresents the information you have, or the estimate you're making, or the risk assessment you're about to make. So the vector index here takes into account the infection rate and the density, or rather the relative abundance of the mosquitoes in the area. So that is a much more accurate method to use, or index to use, if you're going to be, if you're estimating or trying to estimate risk. Now, the other one is determining when humans are at risk. Usually we use thresholds, as it is done in agriculture. If you end up with 3 mosquitoes per trap per day, then you're likely to end up with an outbreak. And also to identify geographic areas of risk, because vector-borne diseases are not uniformly distributed over an area. They occur in foci. When you can identify the foci, then you can put all your efforts into those places, or more of your efforts into those places than anywhere else. Mosquito, if used properly, mosquito surveillance can provide you data for risk assessment in a timely manner. And lastly, number 5, it is to monitor. It can help you monitor the effectiveness of the control methods. In other words, if you're doing surveillance and you say you're adulticiding, you get information of if what you did actually worked, if you see decrease in the number of mosquitoes after you have adulticided. So that is one of monitoring the success. It may not be just the number of mosquitoes, but it may be like decrease in infection rates, or a shift in the age structure of the mosquitoes, or something like that. Now, this one is, I'm going to give you 2 contrasting views. This is a map of the adult traps that we have put in the city of Phoenix. They are showing each of those blue dots is a trap. And you're going to see that they tend to be concentrated in some areas, not in others. The program was put together in such a way to protect human beings. And if you've ever been to Phoenix, you probably know that most of it is desert. So these are put near human residences. Now, this one is a little bit different, because this is the city of Chicago. Now, where you see here, you're going to see that the traps are more or less distributed all over the place, because every area of the city tends to be like a good habitat for the mosquitoes of interest. Now, as it turns out, though, despite the fact that the entire city looks the same, what happens is that most of this was done for West Nile, and most of the activity was in the northwest, that is on the north towards O'Hare, and on the southwest. Interestingly, that is where St. Louis outbreaks took place way back in 1975 and 1976, and the transmission dynamics are pretty much the same, or the transmission system. Culex pipiens mosquitoes and birds. So there's some dynamic there. Nonetheless, getting a map of your trap location is always a good way of finding out how well covered the area you are, and then giving you the information you are going to need to respond, since the distribution of vector-borne diseases is never uniform. Additional functions of a mosquito control program is identifying and mapping larval habitats, because that will give you an early estimate of the future adult abundance, and the information of which larval habitats are the major producers of the adults. And based on that information, you can direct your larviciding operations. And if it is container breeders, you know what to do when it comes to source reduction. Now, this kind of mapping are best done during the winter time, as I'm assuming that we got a winter there, because when all the leaves are down, it exposes all the hidden, or most of the hidden habitats or possible mosquito habitats. So winter time is usually a good time for finding out and mapping the larval habitats, and then you monitor them in the spring and summertime. Also, you can use ovitraps for some of the mosquito species, especially the stegomyia, because it is a good way of finding presence or absence. Also, it gives you the distribution map of some of the species, and it could be used for relative abundance estimates in some of the species, not all of them. Now, whenever you're doing that, this is one of the maps showing the places, the larval sites for, or larval habitats of some of the mosquito species. I took the example from Northwest Cameroon in West Africa. It is just an example, but it always helps. And not only that, this one is really, really detailed, because it tells you where the Culex species are in green. In orange, you see where the Anopheles species are, and the Aedes species in some of those areas. So it is a really good comprehensive map. Now, this is one of the, when you're doing mosquito-borne disease surveillance, this is one of the, some of the aspects that could be used to predict outbreaks in the early season. Climatic factors. We talked about the rainfall or the winds, but there are quite a few other things that could be used. Flooding, runoffs, snowing, and fall and winter temperatures. So there are quite a few that could be used to predict what is going to happen in the future. Unfortunately, this is something we don't do that often, and we don't have, in very few cases have we got really good data on what has been happening before outbreaks. So at least it isn't published, but that kind of information is always important. The mid-season ones is when you start monitoring vector populations, estimates of vertebrate hosts. Mostly this is birds or rodents or any other apart from the humans. And then evidence of virus activity. Usually you expect to, for instance, in the city of Chicago, there was the expectation that you tend to detect the first positive West Nile mosquitoes at the beginning of August. So when you start detecting them in June, then you know you've got a year that may end up being an outbreak year, and you start putting your systems together or to prevent further amplification of the disease. Now, the late season predictors, unfortunately, are those what we tend to use. That is when there's spillage of the pathogen into sentinel birds or epizootic and epidemic vectors, domesticated animals, and so on. So usually this is the one we use, but the other two tend to be ignored. This is what you are going to, you should expect. And I'm using minimum infection rates in here. And actually, I didn't tell you that minimum infection rates and maximum likelihood estimates are always useful to compute, because historic data tends to be stored that way. So for comparison to the historic data, these two indices have got that kind of use. But anyway, this is something you may end up with. When there is no viral activity in the area, 0, then you are not in trouble yet. Between 0.1 and 3.9, viral activity is present. You have just to increase vigilance. It is not outbreak yet. Now, if it is above 4, that is high level, and human cases are likely to be imminent. So this is one of the examples where you can set on a local level in order to guide your operations in that area. Now, don't use this universally. This is something that was determined on a local level. And please, you have to, if you have got a program of your own, you have to determine that. Now, these are some of the other indices that have been used before. Just wanted to show how wide they could be. It was shown, for instance, that a container index of less than 10% and a house index of less than 5% in South America some time back were enough to stop an outbreak of yellow fever. If you don't know what a house index is, that a container index is the percentage of water holding containers infected with larvae or pupae. House index is the percentage of houses infected with larvae or pupae. Now, the Brito index of less than 5 was enough to stop an outbreak of yellow fever in Senegal. The Brito index is the number of positive containers per 100 houses inspected. As you can see, doing mosquito surveillance is labor intensive. You got to do a whole lot of it. Now, I'm not going to go through all of these. I'm just going to, I mentioned two things about the last two, where it is showing that relative abundance of mosquitoes of between 2 and 3 mosquitoes per trap per day using BG Sentinels was enough to stop a dengue outbreak in Puerto Rico. Also, 3 mosquitoes per trap per day or per trap per week using the AGO, that is the Aedes gravid autocidal trap, was enough to stop chikungunya outbreak in Puerto Rico again. So there are different indices that can be used in different areas. Now this one coming back closer to home, what this shows is the vector index here in yellow and the number of human cases in blue. One, it shows that actually the vector index predicts when human activity or human infections are going to be. And so accurately that even if it is bimodal, the human cases were bimodal in this case, so was the vector index. Also, you can use this to end up determining what the threshold is going to be. In other words, the first human case, you go back 2 or 3 weeks, and then you find out what the vector index was at that time, and that will give you the index with the threshold at which people get infected. In this case, it was a little bit more than 0. Actually, there's a mistake there. It should be 0.2. The vector index was 0.2. That is when the first human case showed up. So whenever you're approaching 0.2 vector index, then you have to act because that is the time. Now, using that, you can end up demarcating because you have to do this like on a weekly basis to make sure that you know the vector indices at that very time. So this was August 17 to 23rd, 2007, and it was showing that there was a hotspot a little bit south of Midway Airport in Chicago. So using this kind of data, you can determine where to go and act. Now, this is going to be my pretty much last slide. When you are putting together a mosquito surveillance program, there are 4 things you have to observe. One, it has to be simple. It has to be easy to carry out. Usually in mosquito surveillance, what we do, we use graduate students or seasonal workers. And so it has to be simple enough that they are able to carry it out without making mistakes. Number 2, it has to be economical, cost effective. If it gets to be too expensive, then you cannot sustain it. 3, it has to be effective. However simple or economical it is, it has to be predictive. In other words, you have to get information from it that will predict an outbreak. If you don't, then it is no use having one. In other words, if you're planning for a program to cover, say, the city of Fort Collins, then you're going to need somewhere like between 50 traps well distributed over the city. Then you know that it is going to be effective because you'll be able to sample mosquitoes in all the corners of the city. Now, unfortunately, most cities are much bigger than that. So the bigger it is, the bigger the higher the number of traps. And lastly, it has to be sustainable. It has to be long-term because whatever data you get is compared on the data you got before. In other words, we don't have absolute terms, we have relative terms. So if it is long-term, then you can compare what is going on consistently. And that way you might end up establishing thresholds and determining threshold or areas of higher activity compared to the others, how consistent that is, and so on. Everything that is very useful in predicting and preventing disease outbreaks. Just to a reminder, thresholds must be determined regionally because of what we talked about, the differences in transmission dynamics and the vectors and ecologies of the different areas, and thresholds are determined on basis of historic data. So that is the sustaining of programs gets to be really important. Now, lastly, I'm just going to leave you with one of my favorite mosquitoes. Thank you very much.
Thank you, Dr. Matabe, for showing how the CDC monitors changes in arborvirus transmission dynamics. Next up is Charles Lubelzik, who will take us behind the scenes while trapping mosquitoes in Maine. He will show how the types of traps selected depends on the disease of concern. Take it away, Chuck.
Thank you, Marcia. So our lab at the Maine Medical Center Research Institute works cooperatively with our state agencies, including our Maine Center for Disease Control, on mosquito surveillance in our state. However, we do work across state boundaries, working with folks in New Hampshire and Vermont, coordinating with them, as well as with the federal CDC with people such as Dr. Mutebe. Now, for surveillance programs, it really is going to depend on the disease of concern, and I'm going to focus on the diseases that we are surveying for here in Maine. The first three that you can see on this slide are West Nile virus, Eastern equine encephalitis virus, Jamestown Canyon virus, and then also we are surveying for exotic Aedes species because of the potential of importation of these as vectors of either Zika, dengue virus, or chikungunya virus. Now, in particular, West Nile virus, which has been present and detected in Maine since 2001, is going to target primarily Culex species with a few bridge vector species as well. So for surveillance for West Nile virus, the trap of choice primarily are going to be gravid traps. And here I have a slide delineating both the CDC gravid trap and the Reiter gravid trap. The purpose behind the gravid traps is that they have a container, which you can see on the bottom of each slide, which would contain nutrient-rich water, which would be particularly attractive to gravid or engorged female Culex mosquitoes. These mosquitoes will fly into the water basin to try to lay their eggs during the evening. However, a motor will suck these mosquitoes up into the trap. The benefit of this kind of trap is that these mosquitoes are gravid and have probably fed on potentially viremic positive birds, which would mean you have a greater chance of finding positive virus in these mosquitoes. Now, the other method of surveying for West Nile virus, as Dr. Mutebe mentioned, is a collection of larvae. And because West Nile virus is strongly associated with urban environments, these are just two examples of possible breeding habitat. Now, Culex mosquitoes, particularly Culex pipiens, like nutrient-rich water. So any water body that would have nutrient organic matter, such as leaves or pollen, could potentially serve as breeding sites. However, two that are frequently highlighted in the literature are catch basins, which are a function of a storm water system in many municipalities, as well as gutters. In the early days of the West Nile outbreak in New York City, gutters were found to be particularly adept at housing larval mosquitoes because of the large amount of organic matter which would get trapped and then prevent drainage, which would then allow the mosquitoes to lay their eggs within these containers in upper elevations of buildings. Now, when you are sampling for the Culex mosquitoes, these are primarily the enzootic or amplifying vector for West Nile virus. Now we have Culex pipiens restruens complex, Culex quincofasciatus, and also to some extent Culex salinarius. Now this last mosquito, unlike pipiens restruens or quincofasciatus, salinarius is actually strongly associated with coastlines, and it does breed in saline conditioned water. However, it has been shown to be an amplifying vector. And so in areas along coasts, salinarius should be considered for surveillance as well. Now, the reservoir for West Nile virus are frequently delineated as migratory birds, and many of the Culex mosquitoes are ornithophilic, meaning they feed on birds primarily. The next virus that we are looking at in Maine is Eastern equine encephalitis virus. And again, we target, the enzootic or amplifying vectors along with multiple bridge vectors. Now, for surveillance for Eastern equine encephalitis virus, we have several different methods. One would be the collection of gravid females in resting boxes, questing females in CDC light traps, and then the collection of larvae in forested or wetlands crypts. I will go into each of these in the So for surveillance for gravid females, the primary tool of choice is known as a resting box. And in this slide here, you can see that this is a resting box made of a corrugated plastic, very lightweight and weather resistant, meaning it can stay out for the entire season. Much of the early literature with resting boxes had them being made of composite or wood, which would break down under rain conditions, and also much heavier. So when Dr. Matebe was talking about ease of access of these surveys, one individual can carry many of these lightweight corrugated resting boxes as opposed to the heavier wooden resting boxes. And they have the ability to stay out all season. And so once a good site is found, there doesn't have to be a worry about weather destroying these collection devices. CDC light traps are beneficial for collecting questing females of both the amplifying vector, Culicida melanura, but also many of the bridge vectors, which will transmit Eastern equine encephalitis virus. And in particular, we worry about Cochlearia perturbans, the cattail mosquito, as well as Aedes vexans and Ochlorotatus canadensis as potential bridge vectors, and the CDC light traps, which are baited with CO2 and a small light device, will attract in these mosquitoes during the overnight. Collection of larvae for Culicida melanura is very habitat specific. And Culicida larvae are particularly noted for their ability to use a type of habitat called a forested wetland crypt, which is frequently found, at least in the Northeast, in red maple swamps as well as Atlantic white cedar swamps. Now, in the slide on the right, you can see a small hole at the base of these trees. These are actually holes leading into underground chambers, which may extend down to about 1 meter in length. These holes contain rainwater during the overwinter, which does not freeze, meaning that although you may have surface water freezing hard during the months of January, February, and March, the underwater crypts do not, and the Culicida melanura larvae actually stay active as larvae during this winter season. And so larval surveys can occur from December through March, where the melanura will be active in these forested wetland crypts. Now, Jamestown Canyon virus for us is an emerging disease in Maine. We have had our first cases of it reported in 2017 with a follow-up subsequent case in 2018. Generally, it's believed that this disease is probably under-recognized and has probably been present in Maine for a long time. However, it has come to our attention only recently. So as a consequence, we are performing surveillance for this. We primarily will target several mosquitoes for this virus, including what are called quote-unquote snow pool Aedes or Ochlorotatus mosquitoes, as well as Cochlearia perturbans, and then 2 species of Ochlorotatus that are habituated in salt marsh coastal habitats, and also ones that are occupying woodland pools. So for Jamestown Canyon virus, we primarily rely on CDC light traps. These again collect all of the amplifying vectors as well as bridge vectors that are found and are attracted to this trap. However, there are a variety of habitats that need to be sampled for surveillance for Jamestown Canyon virus. Because of the wide variety of mosquitoes that apparently transmit Jamestown Canyon virus, we worry about everything from salt marsh, coastal habitat, cattail marshes, what would constitute vernal or snow pool water bodies. These are ephemeral water bodies that occur early in the spring into early summer that are around after snow melts. However, these water bodies usually disappear by the beginning of August in the Northeast. And then finally, also habitats such as forested wetlands that are also associated with Eastern equine encephalitis virus. And now finally, exotic Aedes species are a recent addition to our surveillance programs in Maine. This is of course because the presence of Zika virus in 2016 really jump-started a lot of activity to look at the potential distribution of these mosquitoes. And while we have not yet documented them in Maine, they are occurring in at least one species. Aedes albopictus is occurring in northern Massachusetts, not very far from our border. And we suspect that it's only a matter of time before it is found in our state. Now, again, primarily you're going to be surveying for potential breeding locations, and many of these Aedes species, particularly Aegypti or albopictus, are highly adaptable to human mediated environments, and as a consequence they occur frequently in urban environments, and they can breed in very small containers of water, and so targeting areas where you have urban water bodies of stagnant water occurring are going to be highly effective. And so this is an example of many of the different kinds of water bodies that can be found in an urban environment, including but not limited to also solid waste areas where you may have plastic or disposable water bottles that are semi-crushed, but still able to collect rainfall and organic matter for the larvae to feed on. They may as well act as a breeding location for these potential mosquitoes. Now, ovitraps come in a wide variety of sources. These are primarily similar to the gravitraps in many ways, in that you have a container of water, frequently a dark container, which will act as an attractant to the mosquitoes. Mosquitoes will fly in to lay their eggs in this water within the container, and then they become trapped and unable to exit. Now, the ovitraps are used for either surveillance or control. If control is the target, then these ovitraps may be laced with either a material that will catch and hold the mosquito and prevent them from leaving, such as an oil, such as olive oil, or you may also apply insecticide around the inside of these containers. So as the mosquitoes fly around inside, they get exposed to pesticides and die and are not able to exit. So I'm going to talk briefly in the next couple slides about one type of overtrap that we're using called the BG GAT. The BG GAT is used again for both surveillance and control with the two methods I mentioned earlier. And has been found to work for both Aedes aegypti and Aedes albopictus. However, because we do not have as yet albopictus or aegypti occurring in Maine, we have found that they are also useful for sampling for other container breeding mosquitoes, including our exotic Aedes present in Maine. And so for the last couple of years, we've actually been using BG GATs in several urban locations in Maine. And this is a slide with some data presented by Elizabeth Henderson from our lab at the Northeast Mosquito Control Association meeting in December 2018, where we have found that despite the fact that we have no albopictus or aegypti present, Aedes or Ochlorotatus japonicus is easily found in GAT traps in urban locations in Maine. We also, to a lesser extent, have Aedes tricereatus, our native container breeder, present in these traps as well. One interesting thing to note with these use of these overtraps is that populations of japonicus tend to be found earlier in these GAT traps as opposed to traditional traps such as gravid or light traps. Generally, with the use of gravid or light traps, we tend to see japonicus appear towards the end of July, going on into August and September. However, we've been able to find them in late June and early July using the BG GATs, indicating that they're pretty successful and that we feel that if albopictus or aegypti were present in Maine at these sites, we would be finding them.
Thank you, Charles, for your detailed discussion on the selection of mosquito trap types. Next up is Dr. Kathy Murray, who will share her field and lab experiences as she helps to protect public health through mosquito surveillance. Kathy, presentation is all yours.
Well, thanks for having me on. As Chuck and Dr. Mutebi have both given a really comprehensive overview of many of the different kinds of traps and why we do surveillance. I work for the Maine Department of Agriculture, Conservation and Forestry. I'm an entomologist, and we do collaborate across different agencies and organizations within our state and also with the feds and with our counterparts in other states in an effort to protect the public health. We see that as one of our mandates within the state, and many of the entomologists, including myself, that work for the state, we work within the Department of Agriculture, and we're responsible if there was a statewide outbreak and there was a need for a statewide coordinated pesticide control effort to control mosquitoes, that would fall under the purview of the regulatory parts of that would come under with my department also. So in Maine, we have a relatively small number of people compared to many other states. There's 1.3 million permanent residents, but we also have a very large seasonal occupation. Many people have second homes in Maine. We have 37 million visitors a year. You know, we're vacation land. And both the people that live and work in Maine and many of these visitors spend a tremendous amount of time outdoors. Outdoor recreation is really one of our things that we're known for. One of the things that we worry a lot about is eastern equine encephalitis. This map shows, you know, the incidence of neuroinvasive cases. That's the one where it really gets into your brain and infects the brain and causes encephalitis. And many people where it gets to that point die. It's a very serious disease. And as you can see from the map, most of the cases of triple E have been up and down the eastern seaboard. Relatively rare, speaking in comparison to some other kinds of health risks, but because it is so serious, about 1/3 of the people who get it will die. Another third will have serious permanent neurological impairment. So we take it very, very seriously, and we worry quite a bit about it. Eastern equine encephalitis, as the previous speaker mentioned, is largely amplified in birds and carried by a particular species of mosquito that bites birds, becomes infected by the birds, and then that infected mosquito bites a different bird and causes that bird to become infected. And so that cycle of amplifying virus prevalence in the bird population happens in nature, in these songbirds. Occasionally, well, maybe more than occasionally, but the way that humans get it, and horses can also get it, is oftentimes a bridge vector, as previous speaker mentioned, will bite a bird that's infected and then carry it to people or to horses and infects them. And fortunately, we are dead-end hosts. A mosquito that bites us will be unable to carry, bites a person that might have triple E, well, besides the fact that they'd probably be hospitalized and not exposed to any other future mosquitoes. But even if they were bitten by another mosquito, that mosquito would not be able to pick up the infection from them or from horses. The mosquito that is known to be a primary vector among the bird populations and helping to amplify that virus in the bird populations is this mosquito called Culicida melanura. It has a fairly unique larval habitat. As I think Chuck mentioned, the larvae lay their the adults lay their eggs in very acidic water found in hardwood swamps, maybe in the rotted out stumps of trees, in these things called underground crypts, little pockets of water that are exposed just a little bit on the surface and surround the roots of trees in these swampy areas. It feeds at night, and then in the daytime it seeks dark cavities to rest in, and that's the advantage of why these resting boxes work so well, is it provides an artificial resting cavity for these mosquitoes to rest after they've had their blood meal the night before. And as Chuck mentioned, they're made out of, the ones that we use are made out of a weather-resistant corrugated plastic, very lightweight. So we stake them down with a zip tie and a wooden stake to make sure that they don't get carried away by some curious wildlife or blown away in the wind. And we put 10 of them out in each site, about 25 feet apart. We have the opening. These are 1 foot by 1 foot cubic boxes, but they're open on one side, and we face that opening west to take advantage of, well, that it'll look the darkest to the mosquitoes that are looking to for a place to rest at the end of the day. Our sampling season in Maine goes from July 1st until September 30th, unless we have virus showing up and the temperatures haven't dropped down below freezing yet. We may extend it up into October. That doesn't happen every year, but occasionally it does. What we do to get the mosquitoes out of those resting boxes is we use a mechanical battery-operated vacuum aspirator. That method for these resting boxes is described by Kranz, 1989, in that citation that's showing on your screen. About once a week, well, exactly once a week, we visit these resting boxes in the morning. We combine the collections from all 10 of the boxes in any particular site. There is a nylon stocking that's fitted into the end of these, and then that's how we collect the mosquitoes into that nylon stocking. As I mentioned, we collect all the mosquitoes from all 10 boxes just by keeping the motor running between the 10 boxes to help keep those mosquitoes in that stocking, and then take those mosquitoes back to the laboratory. This we're Yankees up here in Maine, so we make our own mechanical aspirator using recycled parts. Chuck gave us one of his old ones, and then one of our colleagues here in the office, who's pretty handy with tools, built us a number of these just out of sheet metal, an inline fan, that plastic collar that goes around the top. We just used another piece of corrugated plastic recycled. You can see the printing on there because it was taken from an old sign or something. It's battery operated. We use a 12-volt battery to power it. It lasts for about 3 sites. In addition, we do, as Chuck mentioned, we use light traps. We just put up one of these in each site. We run those one night a week, and we put dry ice in that cooler that you see above the trap. It's a little beverage cooler. We open the spigot of that so that as the dry ice is sublimating, the CO2 leaks slowly out. There's a small light, and a fan, a vacuum fan underneath that black circular rain cover there that collects mosquitoes overnight. We hang those about a meter and a half off the ground, and run those one night per week. Again, one at each site. Back in the laboratory, so after we finish the collection, the very same day we run back to the lab and cool the mosquitoes down. We keep them alive in a cooler, in that nylon stocking, or in the trap, the light trap container. Keep them alive and cool in a cooler, and take them back to the lab so they're still alive. We keep them cool. We put them on top of, in this case, we're just using, I don't really have a really fancy lab or anything, so we just kind of cobble things together. We have a marble tile that we put on top of dry ice to make it very nice and cold and keep it cold, and then we put that marble tile with the mosquitoes in a watch glass on top of that, and put that underneath a microscope so that we can identify the mosquitoes and keep them very cold, because we don't want the DNA to start to degrade, which will happen if the mosquitoes are dead and then start thawing out. With Culicida mosquitoes, there's a couple of identifying characteristics, but that red arrow that's pointing to a little row of setae, or hairs, on the underside of the wing is quite characteristic, and so we can pretty rapidly identify the mosquitoes that are the Culicida mosquitoes. With the other mosquitoes, it takes us a little bit longer, but we have a few tricks. We use our go-to guide is the Identification Guide to the Mosquitoes of Connecticut, written by the Connecticut Ag Experiment Station folks. It's a really wonderful guide, and that's our primary go-to resource. In addition to the Culicida mosquitoes, we also identify the other vectors that are particularly bridge vectors, and we put those ones that are will be tested for viruses in a sterile little plastic tube using sterile techniques, so that we won't get human DNA on there, or enzymes from our hands that will break down the DNA that's in those mosquitoes. And we take them over to the Health and Environmental Testing Laboratory, which has real-time PCR, and within a couple of days they can test those mosquitoes for us. They put it into a -80 freezer immediately so that it really preserves the DNA, and then almost immediately they can turn that around and let us know, and the Public Health Department know, whether those mosquitoes were positive or negative for viruses. We don't have a really big monitoring program. We wish we could do more, but we're a very small state, and we have and kind of resource poor. But with a good, strong collaboration between Chuck's organization, the Maine Medical Research Institute, my organization, the Department of Agriculture, and sometimes we can get collaboration with some private contractors too. And this map on the right shows that most of our monitoring is done in the southern part of the state, a little bit in the central part of the state, and even a couple of sites way up north and down east. But if you look at the map on the left side of your screen, that shows the human population density. So we're mostly monitoring where the people are mostly at risk for Eastern equine encephalitis and those other vector-borne diseases. In addition, we're trying to be as efficient as we can. And Dr. Mutebi mentioned, mapping is a really good way of trying to identify where the most likely sites are, track mosquitoes and the diseases, and do the best job we can of protecting human health. So in addition, we've also been making an effort to use GIS capabilities and try to identify where the most likely places are for Culicida mosquitoes and some of those, and triple E for that matter, and so that we can most efficiently place our resting boxes and our light traps and plan for if we have an outbreak and where in addition to our regular sampling sites we would want to expand to. As I mentioned, it's definitely a collaboration between all the different groups that work together in Maine. We're fortunate to have a very collaborative group. And that finishes up my presentation.
Kathy, it was great seeing some of the northeastern environments that mosquitoes habituate and learn how to handle mosquitoes in the lab. Finally, we'll travel down to Houston, Texas to hear from Martin Renya, who continues the discussion of the diverse habitats by showing where specific species of mosquitoes are found and the traps that Harris County uses to monitor the mosquitoes. Mr. Renya, the mic is all yours.
Well, thank you very much for the introduction, and thank you very much for the invitation. I'm glad to be here. And so let me just begin by saying that in order to understand and know the type of trap that we want to use to collect mosquitoes and where to set it in a specific location, we need to understand some certain basics about not only the traps, but the mosquitoes and the environment. So an overview of what we're going to see is some environmental factors, some basics for mosquitoes and the types of traps that we deal with, and then I'll enter into what we do here in Harris County. For example, the physiography of Texas. Texas has an elevation that goes from sea level on the coastline on the Gulf of Mexico all the way to over 9,000 to 12,000 feet. And as you can see, they're represented at the top in the Panhandle area and the Trans-Pecos area. The regions that Texas is in is the Atlantic Plains, which covers the Gulf Coast, the interior plains, and the Intermontane Plateau. That's where the cheapest mountains are in the Big Bend National Park in the Trans-Pecos area. So that starts giving you an idea of what the terrain looks like, water availability, whether it's creeks or rivers, etc. And then these regions get divided further into the Gulf Coast, the Great Plains, the interior lowlands, and the Basin and Range provinces. Then the type of climate that we have, it's in Texas, is humid subtropical, all in green, and then hot, cold, semi-arid, and hot, cold desert. The difference between those last two in yellow and in red and pink is basically the humidity. And as you can see, the green, where that humid subtropical climate is due to all the vegetation and all the rivers and creeks that flow into the Gulf of Mexico. The average temperature in Texas is somewhere around 69 Fahrenheit, and the average precipitation is around 34 for all the state of Texas. However, here in the county or the eastern part of Texas, the average can go anywhere from 30 to 50 inches of rain, and the temperature can go all the way to almost 80. Then those regions that I was talking about earlier, which are important in order to know the ecology of mosquitoes or their bionomics and also their distribution patterns, are linked to the type of vegetation that we have or biomes, and it can be divided according to what source you get, into 6 biomes, certain number of ecoregions or subregions. But mainly what I want you to focus now is going into Harris County. And where is Harris County here? As you can see it right there, it goes into the coast and encompasses the Gulf prairies and marshes and the piney woods. The top boundary of the county belongs to the piney woods. And that is basically elm trees, oak trees, woodland areas, and the soil and the water is acidic. And the Gulf prairies is mostly clays. And you will find short and tall grass and brushes along with some hardwood timber. So here's just a picture of what piney woods look like, as I was mentioning, what type of trees you can find in there. And then on the right side, you can see the marshes, the prairies and the marshes. And then just so that you remember the rest of the physiography in Texas, in central Texas, or what we call the hill country, you can find, acacias, mosquitoes, chaparral. And then the northeast, over there by the panhandle, you find the blackland prairies interspersed with timber. And so finally, in the Trans-Pecos area, in the Big Bend National Park over there by El Paso and places like that, you can see all these mountains where the elevation goes all the way to 12,000 feet. So that kind of gives you a picture of what the terrain looks like and the vegetation, etc., of all those environmental factors. So now the other thing to consider is some ecology on the mosquitoes or their bionomics. And I'm not going to go into all that it involves for the bionomics, but if we can at least remember what type of mosquitoes we're looking for, we can categorize them or classify them into by their larval habitat. So we got the floodwater mosquitoes. These ones lay their eggs singly on non-aquatic surfaces, and that's basically because they require a period of dryness. They look for crevices in the mud, that's dry mud, or the soil, to lay their eggs, and then they go and dry out. And then when this mud becomes flooded, then they hatch. Some species of those are Aedes and Cerophora in general. And then we have the container breeding mosquitoes and lay their eggs singly inside containers, especially the type of behavior that Albopictus and Aegypti exhibit, where they like to lay their eggs not on one single container, but in several, in one gonotrophic cycle. So that these are the species that we have for container breeding mosquitoes, amongst other ones, of course. Aedes aegypti, Aedes albopictus, and Aedes tricereatus. And then we have those that lay their eggs singly or in egg rafts directly on the water surface in contact with it. And then water mosquitoes, mainly Anopheles and Culex. Of course, we have other ones, but they do exhibit different behavior. So now another thing to consider is how they enter. Well, for example, if you're trying to collect mosquitoes during winter, then you need to know what stage they're entering a period of the dormancy. And so Aedes and Cerophora in general, they overwinter diapause in the egg stage. Although a note there for Tricereatus, they can enter into diapause in the egg in the larvae stage. Some Coquiletidae and Culicera, they go into larvae stage. Anopheles and Culex, they overwinter in the adult stage. So now, in respect to surveillance, we have three types of surveillance. The egg surveillance, which is the most important because this will give you the most information on the mosquito besides the breeding side. Of course, it is more work that you have to do, time consuming, but it does give you the best entomological kit when you collect the larvae, rear it to the 4 instar, and mount the 4 instar and the exuvi and for a correct identification of the adult specimen. The next one is the larvae and pupae, and then adult surveillance. And for adult surveillance, we have moved into this trend because it's faster and easier to collect mosquitoes. And as long as we have the correct ID on the specimen, then we're good. But so now entering into the adult surveillance, we have two types of mosquitoes, resting population and the flying population. Resting population is those mosquitoes that have just emerged from their pupae into adults, and they're, waiting for their exoskeleton to dry out. And so you can collect those. And also for mosquitoes that just had a blood meal and they're digesting the blood in order for them to start producing eggs. The ones that are looking for protection or shelter, and those that are overwintering. In the flying population, we have those that are looking for nectar, the ones that are looking for a host to feed upon, and those that are seeking mating, and the ones that are ready to lay their eggs. So it is also good to know that what type of trap you're going to use depending on the population you're after. For example, if you're collecting resting mosquitoes, these will give you a more balanced population in terms of their physiological stage, males and females. So you will get unfed, blood-fed, nulliparous, parous, and gravid mosquitoes. And then when you target the flying population, you have to divide it into the host-seeking mosquitoes, and you have to use a trap that is targeting those ones. These traps, for example, the CDC light trap, to mention one, here's an example of the traps for the resting mosquitoes: an underhouse trap, which we use here in the county, red boxes, aspirators, etc. And then just catching up with the host-seeking mosquitoes, other baited traps, CDC light traps, New Jersey light traps, fake prints, etc. All those will give you more species in your collections, but most of them will be young, unfed, and possibly uninfected. That doesn't mean that you will catch some that they're old enough, they have been infected before, and you possibly collect them, but more than likely they will be uninfected. And then we have the gravid or egg-laying mosquitoes. Now here you get the greater likelihood of getting infected mosquitoes, because if they're looking for a place or a substrate, in this case water, to lay their eggs on, that means they already had a blood meal, unless they were autogenous, meaning that they didn't require a blood meal to produce their eggs. But if they're looking to lay their eggs, more than likely they already had a blood meal and they could have been infected while getting that blood source. And these types of mosquitoes are the ones that are ideal for virus surveillance. For obvious reasons. Here's some examples of the traps: gravid traps, rider traps, Fromer trap, the Harris County gravid trap, etc. Let's look into the type of traps. We got basically two lines of traps, one for homeowner use and the other ones that most arbovirus surveillance programs use. Now, homeowners use, they're good. Some are effective, like for example, the mosquito magnet Liberty, it's pretty good, but they have plenty of disadvantages, not only on the attractants that they use, heat, water vapor, CO2, and octanol, but they're bulky, heavy, and very, very expensive. So the types of traps that most of us use are, for example, the OVI traps or the lethal OVI traps, the CDC light trap, CDC gravid trap, EVS, encephalitis virus surveillance, the Wilton trap, fake prints, BG Sentinel, etc. And these use infusions, like for example, what we use here in the county is hay infusion, which is just a mixture of hay and water. The hay is mostly Synodon, and so then we also use CO2. Some of these traps use visual cues, like for example, the base prints, the contrast between black and white, lures, chemical components, so and also light. So now Harris County, located in Texas, you can see it there in the corner at the left at the bottom, and it's by the coast. Harris County is by the coast. As you can see, the elevation of the county goes anywhere from sea level by the coast all the way to up to 300 feet in elevation. And on the map on the right, you can see that there is plenty of water ways here, whether they are bayous, river, creeks, small streams, man-made canals, all flowing into the Gulf of Mexico. So there is plenty of places for good breeding habitats. The next one is, here you can see a picture of Harris County. As you can see, it's heavily urbanized. You would say like urban, suburban, and industrialized areas, mainly in Harris County. So now let's take a look at what we do here in the county. The county is divided into 268 areas, and these areas, we used to trap only for SLE way back when this program started back in 1964 with the SLE outbreak. And so we were using certain types of traps that I'll explain later on, and you will see images of them. But we only had basically New Jersey light traps, and then we move into the sea light traps, but they were placed above ground, hanging from a tree or a light post or something like that. And that was mainly it. Nowadays, we use gravid traps, the CDC light traps placed in the storm sewer systems, and we also use the BG traps. Now, the CDC light traps went from above ground, hanging from a tree, to the storm sewer system because back in 1979, personnel saw a whole bunch of mosquitoes coming out from an inlet, and so they decided, hey, why don't we set a trap in the manhole and see what we get? And sure enough, ever since, over 95% of our collections is Culex quinquefasciatus, the main vector for SLE and West Nile virus here in Harris County. So we started doing that, and that is represented by the yellow areas. And the blue areas, that's where we set gravid traps in people's properties after we get their permission. And the BGs, they are overlaid wherever they ended with, and they were selected based on historical data on the number of Aedes mosquitoes. So we have also used emergence traps. These are designed to collect emerging adult mosquitoes and to monitor the efficacy of treatments in storm sewers and catch basins. And so this is just one type of trap. This is also used in hard to reach places, especially for those float water mosquitoes where you can set it on the mold. We have the floating kind and the non-floating kind. And this is a floating one, but it's put on top of the gutter. This trap, like I said, is placed in a gutter here, but it's useful. So we have also used red boxes, and I don't have to explain about them because you've heard about them. These are mainly they were used in the malaria programs, but as Cathy said, they're pretty good for collecting Culicida. So there's all sorts of red boxes or resting boxes, sticky resting ones, CDC resting trap by different people. All those are used. The New Jersey light trap, pretty good, collecting mosquitoes and some other insects. Disadvantages is that you need an AC outlet and somebody to pay the bill for it. So we moved from there, that type of trap, to the CDC light trap. This is how we use the CDC light trap with the dry ice on that red thermos and hanging from a tree. And then we also do landing raids, especially after disaster events, like hurricanes or tropical storms and whatnot, to do a quick determination of the abundance of mosquitoes. And we also have done some human collections on small projects. And the way we do this, we use aspirators and any kind, whether it's mouth or battery operator aspirators, also vacuum sweepers or backpack sweepers. There's the new BG counter, which is used in conjunction with the BG, and this gives you real-time count of mosquitoes, and you can see the numbers on a web-based page. And so instead of doing landing raids, you can certainly use this type of trap. A picture of the Fay Prince, which uses visual cues, which at least here in the county has outperformed the CDC light trap, but you can carry a lot more CDC light traps, miniature light traps, than Fay Prints in a vehicle. So that's why we don't use those ones. Now, the current collection methods here in the county, as I said, in the middle, the CDC above ground trap, the CDC light trap above ground, we went into the storm sewer cistern right above to the left. We also use the gravity trap on the left and the top, the bottom left, and then the underhouse and the BG Sentinel. The underhouse on the top right corner, and I'll explain about each one of them after this. So here's the underhouse trap. It's a modification of the gravity trap. Here in Houston, there's a lot of pier and beam houses, and so it's a good place for mosquitoes to rest or overwinter. And so whenever we wanted to collect mosquitoes during the winter, we would use this type of trap, and so it wouldn't fit if it was standing like the gravity trap. And so we placed it horizontally, and instead of using water, we use dry ice, and we just push it and pull it out of underneath the houses. CDC light trap, we hang it from the manhole lid with the use of a T-hook. You can see it right in the middle, and then the picture on the right, that's how it looks. And like I mentioned before, over 95% of our collections is quinks. And we still find Albopictus and Aegypti and many other species in here, but obviously in lesser numbers. So now the gravity trap, there's a wider range of gravity trap, the CDC light trap, the rider trap, the Fromer trap. The Fromer updraft trap is pretty good because the collection chamber is placed beneath the motor fan, and so, sorry, before the fan. And so when they are vacuuming to the chamber, they do not go through the motor, so you get better specimens for IDing. This is actually the actual trap that we use here in Harris County. It looks just like the other ones, but it's a little bit different, having that base and that tower and a longer double ring net. Here's a picture of the hay infusion. Ferment these in 32 or 45 gallon trash cans, and we put about 1.3 kilograms of the hay and into 32 or 44 or 42 gallons of water. Then we also use the BG Sentinel traps. On the left side, you have the BG1, which had a lot of issues to prop it and some other issues with it. And then they came out with the BG2 on the right that has the cartridge lure. The first one had the octanol and a lure. The second one has the sweet scent and the cartridge, the lure cartridge. We use it with or without the dry ice to enhance the collections. When we do the dry ice, when we use it, the variety or the diversity of species that we collect is greater. Number one being Culex quinquefasciatus, even in the peri-domestic environment. Here you can see the lure cartridge and the CO2, of course. We don't use the octanol anymore for, it's been discontinued due to EPA regulations. So here's a picture of the attractants. On the left, the octanol discontinued that used to last 3 months, they say, and then the BG2 lure, the sweet scent that lasts 2 months, and the cartridge that lasts 5 months approximately. And now I started mentioning that the best type of trapping or surveillance that we can do is the egg collections. And so back then, Locke in 19, I do not remember, 42 or 72, something like that. They came up with the first lethal ovid trap. And of course, it was used with treated strips attached to the sides of the container. And so they would get killed. As it's been mentioned, we can use it for monitoring or for control. And you can use any type of infusion that you want, regular tap water, a hay infusion, an oak leaf infusion, rabbit pellets, fish food, yeast, anything you want to use. Then, because of resistance to the insecticides, they started moving into a new trend of sticky ovid traps. There, the letter A is the ones with the treated strips, the letter B, sticky trap, the Singapore sticky trap, and the C is the mosquito trap. Then we got the GAD that was pretty successful in Australia on certain studies, and the AGO that was pretty successful in Puerto Rico. These are used to surveillance and for control also. And then finally, here's a picture of the GAD and the AGO, but also the Intucare, which is the newest one in the market, uses an entomopathogenic fungi, which is Valvaria bassiana and pyriproxyfen. And so I'm not going to go into detail on how it helps control mosquito populations, but that's just another one. So once you have put together all these factors, which is the environmental factors, the types of traps that we have, and also the bionomics of the mosquitoes, and you can look at a place, start associating these factors, also the interaction of mosquitoes between them, because certain species associate with several other species. And so when you find one species that you're looking for, usually you will find it associated with other species. So that's a good point of reference. And when you put it all together, you can look at a place like, for example, in the picture on the top left, running water, small stream with slow moving waters, you're more than likely going to find Anopheles, Urantania, just to name a few. On the right, the transient water, you can find Aedes and Serophoras. That's mainly, we don't have that here in the county, but it's a snow pool, right? But we have temporary pools. So, and then when you look at woodland pools, which we have plenty here in the county, more than likely you're going to find plenty of Aedes species. Fresh flood water, if it's rich in nutrients, organic pollutants, you can find quinks, obviously, amongst others, also including Aedes aegypti. Then we got this type of scenario here in the county by the Gulf Coast, tidal floodwater. You're going to find the floodwater mosquitoes, Aedes sollicitans and Aedes tanurhynchus. Then at the bottom, you have those species that look for tree holes, like Aedes tricereatus, among others, freshwater swamps, where you find some Aedes species. And then the bottom picture where you have all those tires, well, you know what type of invasive species we can find, right? Aedes aegypti, Aedes albopictus, and whatnot. When you have acid water swamps like we have in the north border of the county, we can find plenty of Culicida melanura. Well, not melanura because we don't have that species here, but we can find inornata. Pockets of water, we find Culicidas also. And then, like it was mentioned earlier, brackish water, we can find Culex salinarius. And then polluted water, Culex pipiens. If you look under vegetation, those mosquitoes that lay on top of the leaf or the grass, and then they curve their abdomen to lay their eggs on their side of the leaf, you can find Mansonia and Coquiletidia perturbance in cattail swamps too. And then if you look in bromeliads, pitcher plants or bamboo, you can find some species of Wyomia smithae or anything else like that. We do not have Wyomia here, but those are perfect places. Now, let me just say that also you can find quinks and 80 species in these bromeliads. And so if you want results like this or darker, then you need to put all these factors together in order for you to use the right trap at the right place. And with that, if there are any questions, thank you very much. That I leave it up to you.
Thank you, Mr. Reyna, for sharing your many years of mosquito surveillance, management, and lab experiences in South Texas with us.